We are going to talk about genetics and genomics, the basis of evidence-based nursing practice. Our unit objectives are understanding key terminology, discussing various patterns of inheritance, aka genetic mutations, apply concepts of ethics, legality, therapeutic communication to the management of patients undergoing genetic testing or experiencing genetic disorders. So our topics to be covered are going to be the genome, the human genome, and genomic medicine. And this is a very good government link on discussing different aspects of genomes, genomic medicine, the ethical, legal implications of genetic testing and genetic manipulation and treatment. We're also going to be talking about terminology. We'll talk about mutations, patterns of inheritance, and some thoughts on genetic counseling and legal ethical issues. So why the Human Genome Project? A genome is the complete set of genes for a species. So every species has its own genome, and it is the commonalities within that species. So the human genome gives you what your normal genetic makeup should be in terms of lack of mutations and changes. The reason that we do genomic medicine is twofold. We can develop drug therapies targeted to a client's genetic difference. We can also identify patients at risk for health problems for prevention and early detection. Now, some important terminology. Autosomes. Those are your somatic chromosomes. They divide by mitosis. There are 22 pairs. They determine your physical characteristics. They do not determine your gender. The sex chromosomes, otherwise known as germ cells, they divide by meiosis and they create sperm or eggs. So we have a pair of sex chromosomes, either XX or XY. And less than or greater than the normal of 23 pairs is known as aneuploidy. So if you have 23 pairs or 46 chromosomes, that is considered euploidy. If you have less than that or greater than that, then it's called aneuploidy. Some other terminology, allele. Allele is one of the possible alternate forms of gene coding for a trait. So you have alleles for your eye color, for example. You get one from your father, one from your mother. Genotype is the exact allele pair for the given trait. So if you have brown eyes, for example, you could be brown-brown genotype, or you could be brown-blue genotype, because one of the things we know about eye color is brown dominates over blue. The phenotype is the exact observed expression of that trait. So if you have brown eyes, your genotype could be brown-brown or brown-blue, but your phenotype is brown eyes. The other type, some other terminology, homozygous means that you have identical alleles at the chromosome for a specific trait. So your genotype and phenotype are the same. So if you are brown eyes for your genotype, and then your phenotype would be brown. So that is homozygous. Heterozygous, two different alleles at the chromosome for the specific trait. So your genotype, which could be brown-blue, is different from your phenotype, which is brown. So that is heterozygous. And gene expression in this context is that all human cells will contain the gene for all of your traits, but only in specific cells will the trait be expressed. For example, the ability to produce insulin is present in all of your genetic makeup within each cell, but only in the beta cells of the pancreas will that be expressed. The genetic code for your eye color is present in all of your cells, but only in the iris of your eyes is it going to be expressed. Now, this is what's known as a karyotype, and you've seen these displays frequently. And if you look at the very last pair, that 23rd pair, that is your either XX or XY. And you can see in this picture that it's an XX. The chromosomes are the exact same length. So that is the karyotype for a normal female. This is the karyotype for a normal male. So all of them are paired up and they look alike, except our XY at the very bottom. The X is about double the length of the Y chromosome. And that's going to be important when we talk about sex-linked or X-linked mutations. Because if we have a mutation on the X chromosome, which was the most common area where we have mutations in X-linked or sex-linked mutations, there is nothing over here on the Y chromosome that can dominate over it or cancel it out if it's a case of needing two mutation on both cells in both chromosomes in order for that disease process or condition to be expressed. So that is why if we have X-linked chromosomes, frequently those conditions are a higher factor in males than they are in females. And again, we'll talk about that when we get to mutations. DNA is made of 4 bases. We have the thymine and cytosine, and we have adenosine and guanine. Thymine only pairs with adenosine, cytosine only pairs with guanine. And so what happens is, in some situations, some mutations that we're going to talk about, the T pairs with a G instead of an A, or the C pairs with A instead of G. So those are different mutations that can happen when bases do not pair with the appropriate counterpart. So let's talk about the changes. Mutations. Mutations are when we have DNA changes that are passed from one generation to another. So these are inheritances. We can have somatic mutations, which occur on the general body cells, the autosomal cell chromosomes. And then we have the germline or X-linked or sex-linked mutations that occur usually on the X chromosome. Now, there's different types of mutations. There can be a non-disjunction. So during either mitosis or meiosis, the chromosomes do not split. So in one daughter cell, we have 2 of a chromosome instead of 1. And in the other daughter cell, there is nothing. So we have a non-disjunction. So one of those daughter cells will have 1 extra or less than. So monosomy, non-disjunction, an example of that is Turner syndrome, which is at the 45th and 46th, the XX or the XY, and at the split, at the meiosis, that one daughter cell gets an X and the other daughter cell gets nothing. So when they join together again in the zygote, you will not have an XX and an XY, you will have an X and nothing. And that can cause something called Turner syndrome. Trisomy, which you've all probably heard about, Down syndrome, where you have an extra chromosome where the number 21 does not split. So one daughter cell does not have a number 21, the other daughter cell has 2 number 21s, and then when that joins up to the zygote, we have 3 number 21s. That is a trisomy. We can have a point mutation where one base is substituted for another. So a T doesn't pair with A, it pairs with a C or a G. So there's no gaps, it's just a mispairing. Sickle cell anemia is an example of that. Or we can have a frame shift mutation where a base is added or deleted. So we've lost a DNA segment on one strand of the chromosome. And something called Cri-du-Chat syndrome is a frame shift mutation. So let's look at patterns of inheritance, otherwise known as mutations. Autosomal dominant. So with an autosomal dominant mutation, like Huntington's that we talked about in neuro, the mutation is located on an autosome, not an X chromosome, not a sex chromosome. So in the case of autosomal dominant, each child has a 50% chance of getting the mutation. And because it is dominant, they will get the disease or condition that is associated with that mutation. With autosomal dominant, males equal to females because it is on a autosome, not on a sex chromosome. So it's found equally. If neither parent has a mutation, an autosomal dominant mutation, then there is 0% chance that a child will have that mutation, unless something happens when we talk about multifactorial, where something can happen in utero and a mutation can occur. But if in most cases, in the majority of the cases, the autosomal dominant, one of the parents has to have that mutation, and then there is a 50-50 chance for each child of getting that mutation from the parent that has it. So some examples of that, and you can look these up and get some more information about them. Marfan syndrome, Huntington's disease, neurofibromatosis, familial hypercholesterolemia, and something called achondroplasia, a form of dwarfism. So those are things that you can Google and get some information about those if you're interested. Autosomal recessive, the mutation is located on the autosome versus the germ cell, and it can only be expressed if both parents have that recessive mutation, and then both parents would have to donate that mutation to the child in order for the child to have the condition. Because it's recessive, they have to have both recessive mutations in order to have the condition. If they only had one mutation, the mutation from one parent and a normal chromosome from the other, then they would be a quote-unquote carrier of the condition, but not have the disease itself. So it may not appear in all generations. It may skip, and it usually first appears in siblings because the parents are both carriers. They may not have the condition themselves, but they are carriers. So the parents are heterozygous for the mutation. That means they have a normal chromosome and a mutated chromosome. So each child has a 25% chance of being affected, meaning that they have the condition, and they get the mutated gene from both of their parents. They have a 25% chance of being unaffected, meaning they don't get the mutated gene from either of their parents, so they have two normal chromosomes. And they have a 50% chance of being a carrier, which means they get one mutated gene from one parent and a normal gene from the other. And it is again found equally in males and females because it is autosomal, not sex-linked or X-linked. So for examples of that, sickle cell anemia, cystic fibrosis, Tay-Sachs disease, phenylketonuria, PKU. And then another type is your X-linked or sex-linked. And in this case, the mutation is located on the X chromosome. It can be dominant or recessive, but the majority are recessive. And if it is recessive, the incidence is greater in males than females because of what I showed you on that karyotype. The X chromosome is longer than the Y chromosome. So if there's a mutation on the X chromosome, there is less likelihood that there is something on that shorter Y chromosome that can dominate over that recessive mutation on the X. So if the female is a carrier, the sons have a 50% chance of being affected. The female is going to donate an X chromosome to the cause. She's got two X chromosomes, so one of them has the mutation, the other doesn't. Their son is going to get an X chromosome, 50-50 chance of it being the mutated one. The daughters, if the female is a carrier, have a 50-50 chance of being carriers, which mean that they get the X chromosome, one of their X chromosomes is the mutated one from their mother. The affected man who has the condition, so he has a mutated X chromosome, a normal Y chromosome, all of his daughters are going to be carriers because he only has one X chromosome to donate to the XX cause. So all of his daughters are going to be carriers. So some examples of X-linked recessive genetic mutations: hemophilia, red-green color blindness, Fragile X syndrome, which is one of the most common forms of intellectual disability, and autism. Other types of inheritances can be what we call multifactorial, which means that there may be a predisposition to a condition within a family, and then other factors like environment, like diseases that the mother might contract during their pregnancy, that can lead to that genetic mutation being expressed and coming to the forefront. So some of those multifactorial conditions can be cleft lip or palate, pyloric stenosis, club foot, spina bifida, some congenital heart abnormalities. So what's our role in genetic testing? Let's say that you're working at a high-risk pregnancy practice or in a clinic of some sort, and parents come in, and the prospective parents come in, and they want to be tested for specific things that are happening that might be occurring in their family. Maybe someone in their family ends up being diagnosed with Huntington's disease, for example, and they want to find out if they have that genetic mutation, because as an autosomal dominant mutation, they want to know, am I going to get this condition, because this particular condition doesn't manifest until the late kind of end of your childbearing years, versus other types of mutations which appear at birth. Or they may want to find out if they are each carrying that recessive mutation for cystic fibrosis. So our role when it comes to genetic testing is to explain very thoroughly to our patients what that testing involves, what the potential consequences can be if they end up testing positive for a particular condition. And then we have to really spell it out to them what the impact can be on their children. Is this something that can be passed on? Is this something that can only be passed on if we happen to have a child with another person who has that recessive mutation, for example? So it is incumbent upon us to give them the facts without judgment, explain everything as clearly as we possibly can, and hope that they make the quote-unquote right decision, so that a particular disease is not continued on throughout their family and cause heartache and illness and that sort of thing. So it is a very tricky situation to be in as a nurse, to give the facts without being judgmental. And with that, I'm going to close.